I talked too long again. Sorry, guys. Okay, so last little bit, let's wrap it up here. As I was saying, natural selection then could act upon some sort of change in phenotype of a protein, which we had just looked at the hemoglobin example, right? One of the last portions that's in that IQ, the Prezi especially, are going to be the differences between prokaryotic and eukaryotic translation. The basics and the gist of that is that in prokaryotic translation, that can be paired up with transcription because there's no nucleotide because there's no nucleus inside of prokaryotes, right? So transcription and translation can happen side by side right in the cytoplasm with one another. The ribosomes are right next to the genetic code, which is weird, but a benefit of being a prokaryote, those things can pair up. And then polyribosomes can actually happen there too, where you've got an RNA transcript that's hot out of the oven that can just be turned into a polypeptide chain really, really easily and very, very quickly because the ribosomes are right there and multiple ribosomes can attach and do translation at the same time. That does not happen in eukaryotes. Another one of the differences was just what happened after transcription, that there's editing, post-transcriptional editing in eukaryotes, and there's not in prokaryotes. Another thing is that in eukaryotic cells, they're big, they're like metropolises, they're huge cities for cells, which means that their polypeptides, which fold up into proteins, might have to go someplace very, very specific in the cell to do their job, wherever that's located. So there needs to be signal recognition particles, SRPs. I always forget what that stands for, but SRPs are around inside of the cell. And what their job is basically like a little shuttle to take that polypeptide after it's been translated and move it to the correct portion of the cell, wherever its job happens to be, to send it out for packaging in a vesicle and ship it wherever it needs to be within the cell. Okay, so SRPs, that is their job, is to make sure that they're shuffling around proteins in the correct areas. So those are some differences as well. Things that you should be able to do: describe the process of translation in full. Are you able to look at all the important aspects and pieces of what's going on in translation and understand what's going on? Differentiate between prokaryotic and eukaryotic translation, as well as prokaryotic and eukaryotic protein synthesis as a whole. What are the differences? And then interpret the genetic code and use it to determine the amino acid sequence of a polypeptide if you're given the DNA sequence, right? So that's using the codon chart. Are you able to do that whole thing in order to get the correct sequence? Okay, guys. So I look forward to your questions tomorrow and a quick discussion. Thank you.